all of those. And today I am delighted to introduce our seminar speaker, Dr. Nick James, who is he received his PhD in biophysics and biochemistry from the University of Vermont in 2009. And after finishing his training in biophysics as a postdoc at UH Manoa, he became a PI and began to lead research on topics ranging from diabetes to cancer using fluorescence-based applications. He's a biophysicist and eager to apply fluorescent techniques to probe the mechanisms that drive a number of diseases, Parkinson's, Alzheimer's, and diabetes pathologies. And his lab uses several tools for their research, including spectrophotometers, fluorimeters, microscopes, including a 2-photon laser, and genetic engineering. He's passionate about science and committed to helping shape excellent scientists. And he serves as a fellowship mentor for the Hawaii Data Science Institute. So his topic today, the title of his seminar today is Characterization of Metabolically Distinct and Unique Cellular Signaling Populations of Adipose Tissue Using FLIM Fluctuation Microscopy. Welcome, Nick. Glad to have you.
Well, thank you for the introduction, and thank you for having me and everyone who is here listening. So like Gwen said, my seminar is going to focus on my research, which has been characterizing adipose tissue by looking at endogenous fluorescent markers to basically design molecular fingerprints so we can isolate subpopulations and basically be able to then determine downstream alterations like disease states or diabetes formation to try to prevent that. So although Gwen did give a little bit of my background, I kind of listed some here just again to highlight my research interests. I graduated PhD in 2009. That research really focused on protein biochemistry. And then when I joined UH Manoa as a postdoc, my main focus was basically to learn everything I could about fluorescence and fluorescence methodologies, which led to a lot of research being developed in terms of method development. And then as I progressed forward into my PI position as assistant professor, I began to focus on specific diseases that, again, involved my passion, which was proteins and proteins interacting with things like membranes or other proteins. But as you can see, there's always one underlying theme of my research. I basically fell in love with fluorescence as an undergraduate when I did a summer undergraduate research program. And since then, I've just wanted to basically utilize that method, know everything about it, and try to apply it to every system that I've ever worked with. So the question becomes then, why did I fall in love with fluorescence? Well, first of all, it's very pretty, right? As you can see here, you're looking at excitation of different types of dyes, and you can see the range, spectral properties from blue, green, all the way up to red. However, the application of this method to biological systems is also very fascinating. It provides information on molecular environment. So if you have a fluorescent probe, it can become sensitive to things like viscosity, polarity, pH, ions, etc. So you're able to detect microenvironments where the fluorescent probe is located and be able to determine specific processes that are happening around there. Another important aspect of it is that it provides information on a nanosecond timescale. It's important because there's a lot of events that are happening during this timescale, so you're able to characterize a lot of things such as protein-protein interaction or movement of molecules within a cell. And so the way that we look at fluorescent probes and how we use them in cells, that they function basically as a molecular stopwatch, in that you're able to identify dynamic events occurring during the excited state lifetime, or how long before the fluorophore is going to emit, which we call like the public domain. So, and we're fortunate enough that a lot of the processes that we as biological scientists like to study occur within this time range. So those of you that aren't familiar with it, I briefly just give like the briefest background on fluorescence. So what is fluorescence? It is the light emitted by an atom or molecule after a finite duration subsequent to the absorption of electromagnetic energy. It's my favorite definition of it. And so what this looks like in practice is, as you see on the left, there is what they call a Perrin-Jablonski energy diagram. And if you have a molecule that has electrons that are resting within a specific ground state, upon absorption of energy, specifically light, the electron will then get promoted to an excited state. It will then relax down to the lowest vibrational energy level. This occurs within femtoseconds. And because there's such a large enough gap between the first vibrational level and the ground state, see there's a huge energy transition that comes out here. The release of the energy can either occur through loss to the system, like heat, or it can emit a photon at a lower wavelength or a higher wavelength, lower energy as the one that was absorbed. So the right, this is basically just mapping out the excitation and emission spectra. Excitation spectra is fairly similar to absorption spectra. So if you're familiar with that, that's basically what the excitation spectra is. So in this region, it would just, you can excite a fluorophore here, anywhere along the excitation spectra. It'll then promote the electron to an excited state, at which point you'll then end up getting emission. As you can see here, the emission spectra, which will occur over a fairly broad wavelength. Now, because fluorescence is quantized in nature, and we know the things that are possible to change the fluorescence intensity or properties associated with it, we've been able to, over the years, come up with simulations as to different events that we'd be interested in, things like protein interactions, protein movement within cells, things like that. And I'm going to show you a simulation of what this would look like in a specific system. So if we were looking at just, say, a free-floating, like a monomer within like a cell, you're able to then simulate how the diffusion would occur over time. This would be your excitation path. There's molecules just moving into and out of it. And based on that movement, we're able to determine physical properties of the system, things like concentration as well as diffusion. Now, we're also able to simulate what would happen in a situation where you have protein-protein interactions or association with slower moving vesicles. So here, if we just change the parameters a little bit, you'll see that now the diffusion has significantly changed. The rate is significantly slower. And this is based on because now it's interacting with multiple proteins. So again, we would expect it to be a bit slower. So now that's just a simplified case, just protein, again, just proteins interacting with like similar things like proteins themselves or vesicles. But what we really want to get to the point is we want to get to where we can simulate what's happening within inside a cell. So this is just an example of some live cell data that we've taken before. And you see there's a molecule that's moving, a vesicle that's moving, interacting with a specific spot, possibly an endosome, and then progressing on. So again, this makes it much more difficult in terms of simulating such data, as now we have multiple different aspects of the cell. Again, the cell's more complex than like a solution or a single point. So we got to think about different spots, so spaces in the cell, and like things like endosomes or the trans-Golgi network. We have to think about localization of just vesicles as well and how they're going to progress. And then we got to think about the time. So this is basically what, in terms of the fluorescence field, this is how we've been building up to this point. We're able to collect things, we're able to analyze it, and basically make predictions based on simulations as to what might be happening. Now, is it possible to expand beyond just a cell that is in homeostasis? Can we expand into a system where we're seeing disease progression using these fluorescence methods? And the answer is yes. My mentor, Enrico Gattone, has developed several patents to try to analyze these things. So I'll give you two specific examples. So one of them is to basically discriminate between metabolic states. In this case, he was able to look at progression of cancer in live systems, just using endogenous markers, so you don't have to tag anything. And basically, you're able to do that because it can't, under situations where a cell becomes cancerous, the metabolic properties are drastically different than those that are non-cancerous. So we can then determine simply where are we seeing large alterations in metabolic activity, and then begin to pinpoint where or what type of cancer occurs. One example that he did and patented and published was on looking at breast cancer development. Another example of this is something that you're probably all fairly familiar with. It's characterizing disease progression within retinal tissue. So if you've ever been to the eye doctor before and they put that dye solution in your eye to try to characterize it, this is a fluorescent marker, and it's called fluorescein, and they're basically looking at differences in terms of the intensity and concentration of it, because this will drastically alter the fluorescent properties, and you're able to basically analyze how these alterations are occurring to say, okay, this is how far, or this is what type of disease we're undergoing, how it's progressing. So for my research, I'm trying to expand what we've been doing recently with these metabolic analysis and simulations to characterize alterations in adipose function as a predictor of metabolic disease development, things like diabetes. So I'm going to give you the briefest background on adipose tissue function. So again, adipose tissue, there's two different types. There is your white as well as your brown, and within the white, there's multiple populations. So this is your visceral versus subcutaneous. And so adipose tissue, that's our fat, right? And the main purpose or function of adipose tissue, something that you're probably already all familiar with, is the fact that it's involved in regulating whole body energy homeostasis. So under conditions where you have excess energy, the adipose tissue will then convert this excess energy in the form of glucose into lipid droplets that could be used later. And then under states where you have low energy concentrations, the adipose tissue will then activate and break down these lipid droplets. So then that energy can then be transported throughout the different tissue types. Within adipose tissue, there are multiple cells, as you probably can guess. It's not just adipose tissues. There are several different types that are involved in maintaining the health of it, such as like macrophages, endothelial cells, and things like that. And then you also have your precursor cells, your pre-adipocytes, which then can be differentiated into your mature adipocytes. Now, beyond energy storage and energy release, adipose tissue serves to signal other molecules about the energy state of the whole system. So adipose tissue secretes several different hormones, cytokines, and metabolites, or specifically adipokines, so molecules that are specific to adipose tissue. These are key signaling molecules to control things like the central nervous system, as well as metabolic activity of other tissue types. For example, I put just two common adipokines that you're probably familiar with, which is leptin. This is a molecule that controls appetite through signaling within like the hippocampus to basically stimulate satiation. So it basically tells you, okay, I'm full, so I don't want to eat anymore. And then there's adiponectin. This is a key molecule in terms of muscle activity, in that it sensitizes muscles to uptake glucose, so sensitizing it to insulin, as well as stimulating fatty acid metabolism of the muscle. So basically, it's a key molecule to allow for food consumption within muscle tissue. Now, why I'm interested, or why I've been trying to utilize adipose tissue as my system of choice for basically determining or predicting disease progression, is that there are a lot of changes associated with adipose tissue under disease state, which then disrupts several different tissue types and basically disrupts their ability to maintain energy homeostasis. So basically, when you are under sort of a disease state condition like inflammation, you will have disruption to adipose tissue uptake of glucose through developing insulin resistance. Increased inflammation also causes increased in swelling of adipose tissue, which then causes them to end up dying. And then when they die, they will also, again, increase the inflammation and signaling through the system, as well as signaling to other tissues. And there's also significant disruption of other tissue functions because now they're not performing their normal function. You're not getting the normal release of hormones and cytokines to signal to other tissue types that they need to be sensitive to glucose uptake, things like that. So when you have disruption in adipocyte function and you lose that adipokine release, you see that you'll undergo things like increase in vascular tone and the vesical or arterial stiffness. So basically you're getting hardening of the blood vessels. You'll also have things like liver inflammation, which is again going to disrupt its ability to process basically fats and other molecules to be released, as well as you're going to lose insulin sensitivity and muscle perfusion. So your muscles are not going to be able to undergo the uptake of the normal uptake of glucose and breakdown of it. So you're going to lose energy through that way as well. This dysfunction occurs because of, and not only adipocyte alteration, but also the cellular makeup of the adipose tissue is disrupted. When, again, some of the molecules are involved in maintaining their health, things like monocytes and macrophages. And when you end up having a disruption or dysfunction, you see that there's a shift from this anti-inflammatory to a pro-inflammatory state. And this continued pro-inflammatory state is going to be what causes significant disruption to the adipose tissue health, as well as the downstream signal associated with adipose tissue. So for my research, I want to be able to characterize adipose tissue function in a live system. So what would be great would be to take the entire system and be able to do it in humans and measure the health of their adipose tissue, and then basically start detecting if there's going to be dysfunction and try to help develop a personalized medicine for said individuals. To do that, then we can't just be, genetically modifying these humans with fluorescent markers, things like that. So we have to come up with some sort of fluorescent marker to be able to measure alterations in the metabolic activity. And the way my research has been looking at changes in intrinsic markers, such as NADH and FAD, of metabolic activity of cells. So NAD plus, is converted to NADH during the TCA cycle, fatty acid oxidation. NADH then serves as a hydroxide donor for ATP synthesis within the mitochondria. And as you learned way back when in biochemistry, the NADH is then going to serve through complexes 1, 3, and 4. And with that, you're going to produce the most amount of ATP. You're going to end up getting 34 ATPs through this process, versus if you were just going through the normal non-mitochondrial consumption of glucose, you would only end up getting 2 ATP. So how then can we use NADH as a intrinsic marker? Well, fortunately for us, NADH has a unique absorbance spectra or excitation spectra compared to that of NAD plus, and as well as other intrinsic markers there. With the proton bound, you see that there is a increased absorption spectra, or a new spectra appearing at around the 340 wavelength. That allows us to have a selective excitation for just monitoring NADH. Now, with just measuring NADH fluorescence alone, this wouldn't be very useful. However, it was determined back in the 70s that there is a difference in bound NADH when it's associated with a protein versus when it's free. When it's ended up bound to a specific protein like LDH or something like that, there is nearly 100-fold increase in that intensity, as well as the fluorescent lifetime. The lifetime being how long that electron will remain in the excited state before it returns back to the ground state. So now we have a very distinct marker that we can measure as a basically a signal to tell us about the metabolic state of a cell. Now, again, if we want to go into this as like looking into applying this in the real world, how are we then going to be able to measure adipose tissues through the tissue? Well, the advancement of laser technology has allowed us to develop laser systems that instead of using a 1-photon excitation, we can utilize something called the 2-photon excitation. Now, the 1-photon excitation means you just need 1 photon to hit your atom to promote it to the excited state. This is much higher energy and is not very specific in terms of where you are exciting. So in these systems, we used confocal microscope before, the way you actually get your focus on your specific spot is through pinholes. However, with 2-photon excitation, what you have to have is enough photons hitting the system wherein you have 2 photons that simultaneously hit your fluorescent marker to then promote it to the excited state. And then you can end up seeing the normal fluorescence you would see is just like a 1-photon system. And as you see here in this diagram, the bottom left, you see that this is intrinsically confocal because you'll only have enough photons to promote or excite your sample when you have all those photons there at your focal spot. So it's only going to be within your plane of focus. So you see there's very little outside or out of focus fluorescence occurring. Just instead, there's only fluorescence intensity occurring from just the focal spot. What's useful about utilizing 2-photon technology is it also has a higher depth of penetration. So thinking about 2-photon excitation, we're thinking about 900 to like 1,000 nanometers. Again, the depth of penetration is dependent upon the wavelength that you utilize. So there's less scattering or absorption of light the further out you go into the infrared. Probably familiar with this, with when you go to measure, you measure like the blood, the blood pressure, things like that, or your rate. So then they put that clamp on you, and it usually has a red light. That's because you're getting less scatter through the system, so the red light's able to penetrate more, and so they're able to specifically focus on and looking at things within the tissue. So again, you can see here with a 2-photon system, we're able to have very high depth penetration, and with that reduced signal to noise, there's no out of focus excitation or scattering. We can have fairly deep penetration into the system and very focused excitation of our samples of interest. You see there's an example on the right side here where they're looking at neurons, and they're able to have specific penetration and excitation of their system. I mean, it gets a little bit diffuse down at 2 millimeters, but you see out to around 1 millimeter, 1.5 millimeter, you still get fairly specific excitation. So we can utilize like a 2-photon system to penetrate through the tissue and specifically excite only the adipose tissue to collect this metabolic information. Now, how we analyze this is going to be key in terms of determining the alterations within the metabolic properties. Intensity is extremely difficult to utilize as an indicator because it is dependent upon the concentration of your fluorophore within whatever system you're working in. So there could be some that have higher NADH bound, some that are lower, et cetera, and you're not able to distinguish what the alteration is or if there is an alteration within the metabolic activity. Instead, we're going to be looking at the lifetime of the NADH instead. So again, I mentioned there's a significant increase in the lifetime of NADH when it's bound versus free, and we can utilize that to our advantage. So on this sample here, we have a figure just showing the intensity profile of the cells that's been transfected. You can try to extract the actual lifetime information from the system, however, extremely complex because it requires a high number of photons hitting the detector, or what we call counts. Instead of doing that, we utilize a different method, which is called the Phasor analysis. It just takes the raw lifetime data of the system and then plots it on the semicircle. Okay, and when you utilize this system, what's nice about it is that it's basically just a simple algebraic solution if you're looking at multiple populations, so bound versus free. It's just a simple linear subtraction to be able to determine concentrations or contributions from bound versus free. And in this example, they just show how it was more difficult to try to isolate fret, we were talking to Gwen about earlier, between two systems, trying to isolate the actual lifetime data or the lifetime values in the nanoseconds using this one system versus using the Phasor analysis because it doesn't require as many counts to actually extract your data. So how are we then going to utilize NADH FLIM within our system to determine metabolic alterations? Well, within the Phasor system, you see that when you have free NADH, has a lifetime of about 4 nanoseconds. It's located here on that semicircle. When you have bound NADH, it's located at about 3.4 nanoseconds all the way up here. So if we have a system, for example, here they had some cell types within this paper, and then they were looking at the location of the NADH FLIM, they're able to then draw a line between these two, separate out the concentration or the how close it is to the free or how close it is to the bound to determine the concentration contribution from bound versus free. Now, under systems that have more bound, this is when it's going to be interacting with mitochondrial complexes. So when there's an increase in bound NADH, this is your oxidizing condition. So this would be oxidative phosphorylation. So you'd assume the system was fairly healthy. Whereas if you had you saw a change, a significant change where you had a reduction in the reduction in the bound concentration and a shift towards more free, you would say this is more reducing conditions. So they're forming more glycolysis. So there's less mitochondrial contribution, less energy to the system. So we're able to then basically look at concentration of bound versus free and then determine what is the health state of tissue types, or specifically in our case, adipose tissue. And so this is an example of what our data looks like. Oh, that's a typo right there, but okay. Where I was looking at, can we see a difference between the cell types within adipose tissue? So we're looking at pre-adipocytes versus differentiated adipocytes. Is there a difference within their Phasor locations, and can we say something about the system as we're differentiating them under healthy homeostasis conditions? And just the top figure just shows you going from pre-adipocytes to all the way day 12, that's fully differentiated adipocytes. And what you see is that the black circle represents where the Phasor trajectory is with your pre-adipocytes, and the green is the Phasor trajectory associated with your fully differentiated adipocytes. This basically produces a metabolic trajectory that indicates that the NAD population is less protein bound under fully differentiated conditions. Therefore, there's a decrease in oxidative phosphorylation. And we are able to quantitate that, but I didn't put that figure in here, so that's unfortunate. But basically, from this figure, after once you draw the line and determine the concentrations, you end up seeing that there's almost about a 20 to 30% shift in the NADH population, indicating a significant reduction in ATP production of the differentiated adipocytes, which correlates with what has been reported under utilizing other conditions or other techniques. Now, healthy adipocytes have sensitivity to specific cytokines and adipokines. And so we also want to see how these differentiated adipocytes would be would basically have if they would have a shift associated with them when we stimulated them with specific adipokines. FGF21 is a really hot target because it is an insulin insensitive marker or treatment that can be utilized to increase the metabolic output of adipocytes. So it is seen as something that can help with individuals with diabetes by stimulating adipocytes, then which should then help increase their sensitivity to insulin, and basically try to get the adipocytes to have their full functionality again and to prevent other comorbidities with diabetes. So what should happen then is if it's more responsive at increasing ATP production when treated with adipocytes, there should be that shift back or shift towards the to the left on the Phasor trajectory, which would tell us that there's an increase in the bound NADH. Under conditions where we just look at ATP production, there is a significant increase in ATP production when stimulated with FGF21. And then when we did our FLIM analysis of it, we were able to see that yes, indeed, there is a significant increase in the concentration of the bound versus the free. So we're able to then take these Phasor trajectories and then basically state this is where healthy stimulated adipocytes should be located under a tissue under like a tissue type system. We also see that insulin resistant conditions produce reduction in the metabolic activity of adipocytes. So if you overexpose the cells to FGF21, you have a significant reduction in the ATP production and a shift within the fluorescence Phasor trajectory. And this is also seen under conditions of inflammation. So inflammatory or chronic inflammatory conditions within adipocyte adipose tissue is predicted to be a key cause of insulin resistance, then obviously causing downstream signaling, which would then induce things like diabetes or other metabolic disorders. When we have treatment with these inflammatory markers like TNF-alpha or oxidative LDL, there's a significant reduction in ATP production and respiration, as well as a distinct Phasor trajectory for these adipocyte cell types. So what has that provided us? This has basically provided us with a unique metabolic fingerprint of pre-adipocytes, healthy adipocytes, and those undergoing things like inflammatory conditions or disease state conditions. So you can see there's a unique distribution between these different cell types, so we can separate them. So how can we use that then when we're basically looking at human state? Because again, we want to take this information and be able to utilize it in terms of personalized medicine. So how can we then take that? We can then utilize these metabolic fingerprints and utilize simulations of disease progression to then basically track an individual's health and then prescribe them with specific pharmaceutical intervention to prevent further progression of the disease. So basically, the hope is that, or the plan is that, with these mapping of these different metabolic tissues, we can take a unique instrumentation and then excite tissue in vivo that would be non-invasive, and then be able to map out the metabolic properties associated of the adipose tissue and be able to isolate specific regions of this adipose tissue and determine, is there progression of a disease state? Are we seeing reduction in metabolic activity? And how, if so, how can we treat this individual to prevent further progression towards diabetes? So something would look like this. If we were just an example, if we were looking at an individual, we saw an individual's adipose tissue metabolic activity up in this region here, we would say your adipose tissue is healthy, everything you're doing is fine, so keep going, keep doing what you're doing. But then if we had an individual that came in where we saw a metabolic fingerprint in this region here, we could then design treatments, things like treating with metformin or something like that, to basically prevent further progression of reduction in metabolic activity within the adipocytes, as well as other, targeted therapeutics for this individual. We can also then, knowing how alterations in adipocytes can disrupt other tissues, can also then simulate other disruption of other tissue types like cardiovascular or brain or the central nervous system when these individuals are undergoing these disease state adipose tissue metabolic activity, because again, you're going to see a reduction in the release of specific adipokines, stuff like that. So knowing how they're affected by this disruption, we can further simulate how this could be affecting specific individuals, and then again prescribe them not just only treatments for their adipose tissue, but then also try to upregulate the activity of things like cardiovascular tissue or parts of the central nervous system. And another way that we've been able to utilize this technology is by increasing the health and of oocytes, and as well as fetal development. So one issue with reproductive technologies is that you freeze several oocytes, but you don't know what the health or metabolic activity of these individual oocytes are. However, we can utilize these FLIM, NADH FLIM technologies to then track or characterize individual oocytes and to select the ones that seem to be the healthiest or active in this case. And we can also then track the health and development of the fetus utilizing, again, this 2-photon, 3-photon technology, and look at how there's alterations in terms of free versus bound NADH, and determine if there are issues associated with the fetal development. And with these technologies, they've actually seen significant improvement in terms of implantation as well as pregnancy rates and being able to carry on the full term. I still have some time left, and there is it a question in the chat? No. The one last sort of section that I wanted to leave you with was looking at being able to expand further just upon the metabolic activity of the cells, in that we can look at specific alterations in protein expression to make, again, the treatment more targeted. So under conditions of obesity, diabetes, inflammatory conditions, there are a number of protein markers that are involved in maintaining metabolic homeostasis within adipocytes that are significantly reduced. So one thing that I've also been trying to do is track the changes in protein dynamics and expression of these cell types, and to determine how specific treatments can then cause recovery within these systems. Right, so I probably shouldn't go through the theory too much of this, but that's basically explaining what we're trying to do. I don't want to skip too far ahead, but basically we're going to utilize advanced fluorescence fluctuation methods to track the specific protein markers to basically, again, determine how things like type 2 diabetes affects specific pathways, and if these pathways are disrupted to the point where we can't recover them with specific interventions. And again, this is just kind of describing the methodology. We use a circular scanning system around your fluorescent system. It then has a feedback system where it then recovers and tracks the progression of it. And what that looks like is that once, when you have a system and as it progresses in 3D space, it's going to basically hit or come in near contact with your excitation. That's going to cause an alteration within your fluorescent signal. So then optoelectronics will then move to try to recenter the system. And then at the end, you'll get a specific trace that looks like this, telling you how things have moved and where things have moved inside the cell. And so we've been utilizing these methods to look at disruption in terms of protein signaling. And one of them we've been looking at is just common growth factors and how these are disrupted during a disease state. And we've been characterizing the FGF system and how there are alterations in terms of signaling to the receptors to these growth factors, and demonstrating how there is unique phosphorylation patterns that are distinguishable between healthy versus undergoing disease states, and these specific phosphorylation types disrupt FGF2 and FGF21 signaling within your cell, within the adipocytes. There is also significant alterations in terms of membrane expression, and which ends up reducing the sensitivity of the cell type to the growth factors. And there's specific intracellular localization of these receptor complexes under disease state. In this example, I'm showing you that there is, under disease state, there is significant correlation of FGFR1C within the nucleus under the disease state, which indicates it might be serving as a transcription factor or signaling transcription factors and altering protein expression. Again, conclusions of the research is to try to use fluorescence methods to collect metabolic data, to develop systems that can be used in humans to create personalized medicine, basically track disease progression and determine how, with this data, you can slow or even mitigate the disease progression. And we've also been able to do things like increase successful oocyte fetal development, as well as increasing targeted treatment through understanding of protein expression and alterations in signaling within cell types during disease state. I wanted to list out that the resources that I used for the analysis pretty much always use something called SimSDS. It's developed and coded by my mentor, Enrico Bertone at UCI. It is available. There is a copy of it that's freely available on its website too. And the instrumentation and data collection all pretty much occurred all here at University of Hawaii. People are interested. There are things that can do FLIM, particle tracking, things like that, membrane analysis, as well as multiple confocals that you can use from cores as well. And of course, it's got acknowledgements like the Diabetes Research Center, all those in charge, like Mariana Gershenson, who's a PI, internal mentors, and lots of different contributors to this project. So at this point, I'll take any question.
Okay. All right, let's see.
Longer than I expected. I think I just talked too much.
Oh, no, that was great. Okay, how about some questions for our speaker today? So Nick, maybe I have a question, if that's okay. Maybe you mentioned this in your seminar, but do you see this as a possible, diagnostic tool for early stage, say, type 2 diabetes?
That's kind of what the hope is. It's what I'm collaborating with my mentor Enrico to develop some sort of instrumentation that can just quickly scan across adipose tissue, and then basically, again, with the metabolic fingerprints that we have, determine, are these individuals trending towards a disease state? Are they going to become insulin resistant? Then again, we can then again prescribe things like metformin, then check if there's a certain number of time that's passed, be like, how is this affecting the their metabolic health, or do we need to alter our treatment as well to, again, get them back to a point where we're not going to end up seeing insulin resistance and then progression to diabetes? So yeah, that's kind of where we're hoping to take it.
And is that, would those treatments also be applicable to those that have type 1, but maybe trying to control their disease progression?
Yeah, that's a great question. Didn't think about it, but yes, absolutely. Because again, it's just looking at metabolic properties of the adipose tissue. And again, these individuals, they're just born with insensitivity to insulin, but you could track these individuals and look at their adipose tissue metabolic properties over time. And again, you could see maybe something is not working. and they're trending towards an even worse state, and then try to have some sort of altered intervention, pharmaceutical or something like that, to make sure that you can try to stay at your healthy baseline of adipose tissue metabolic activity. Yeah, for sure.
Yeah, great, cool. Okay, how about some other questions?
I was going to ask, how many samples are required to kind of derive the fingerprints that you're kind of alluding to?
A lot. So we use imaging dishes and condition media and things like that. And it's basically one imaging dish is 1 point. And so you collect 10, 20 cells per condition, and then that's 1 point, and then you just collect more and more. For mine, it was almost 200 conditions that we measured, and we just kept going and going just to make sure. Because again, you know, you got to, there are going to be differences in the differentiation. Some might not be fully differentiated, yet you collect them. So there's going to be that sort of plus minus. You got to make sure you get that full range, what you expect to be correct. So yeah, it was a lot. It was like almost a year's worth just to get that metabolic map.
Okay. Are these these are in vitro samples?
Yeah, so these are in vitro. So with them, we've now started to get things like primary tissue. Those things are more expensive, much harder to work with. Now we're starting to get that, use that, and compare them to the map that we have. And again, you can get adipose tissue from healthy individuals, obese individuals, individuals with diabetes, type 1, type 2, things like that. And so we can have that map and fully make it a little bit more defined for these disease states, and then take it into the human condition, while at the same time we're developing the instrumentation so we can actually accurately measure the adipose tissue. Because again, the ones that we are using are more are really clean. They're like pre-adipocytes versus differentiated adipocytes, versus when you have adipose tissue, it's going to have macrophages and monocytes, things like that. So you have to get a map for each and every one of them. And I've collaborated with people, with other research like Mike Corley, and he was looking at monocyte macrophage differentiation under different conditions, and versus healthy versus diabetes condition. And we were able to collect those ones, add them to the map, so that way you can then pull back, find that figure here, you can basically grab like along the Phasor plot here, say this is where my healthy adipocytes would be, this is where monocytes, macrophages would be. You basically would be able to create your cellular map with those different fingerprints. Yeah.
Okay. Wow. Is so the figure images you have there, is that from the software that you and your mentor did?
Right. Exactly. Yeah. And this is actually tissue type that he took from a liver, and he basically scanned through the whole liver, and then he already had the distribution of cell types and alterations. And that's why he's able to highlight the different regions, saying these are the different cellular populations and alterations in metabolic properties within it. Yeah.
So are you guys looking at potentially like a library across groups?
Right, exactly.
Of researchers to kind of like build a big database?
Yeah, and that was one of the things too that I really wanted to test, was like, is there differences between populations? You know, where I could look at the Asian population metabolic activity different from, you know, Caucasian or Polynesian. That was one of my first sort of proposals to do when I put this together, but I got some, you know, pushback, because it's like, yeah, it's not really easy to work with it. You have to have people who are, you know, like medical doctors that are that you know the population trusts and things like that. So it's a little more difficult to get tissue and things like that. But yes, that is something that I'm trying to do as well.
Okay, that sounds awesome.
So Nick, I have a not so serious question. So when you're probably aware of the company Theranos, the
Oh God, yeah.
scandal. So when you first heard that they had come up with a blood test to the one blood test to rule them all, I mean, how much did you think that was fact versus fiction, and what are your general thoughts?
My thoughts on that was like, okay, well, they've developed something. Maybe they do have something. Maybe she just knew exactly what you needed to do to get all that information. But at the same time, mean, you got to be skeptical, right? From that, it was just like a single blood drop. There's so many different experiments that you need to do to get that calibration, and you just don't have the volume to do it. I mean, I looked at her little mini system. Some of the things were like absorption, fluorescence, the things I'm like, that's going to eat up all your sample right there. And you know, for us, for me, the good thing is I'm working with my mentor who's developed dozens upon dozens of patents, and he's even developed one with a system similar to what I want to work with, where you can just basically analyze through the tissue type and detect cancer development. Like I said, he was able to utilize it to develop breast cancer development based just on the metabolic properties associated with tissue. Because again, cancer tissue is or cancer cells significantly different than those undergoing normal homeostasis. So the technology is there, it just has to get expanded for us. And again, also tested to make sure we're not causing any injury and or pain to the system, like the individual, you know. Yeah.
Thanks.
Mm-hmm.
Nice talk, Nick.
Oh, thank you.
Are you able to get video with this?
Mm-hmm. Yeah, you can absolutely. The problem is, you know, the files are so huge, but in the case of what we would want to do, you would have to this final scan, again, it's a 3D rendering, but it is basically just video over time, how things are changing. And I wish I could pull some up to show you what they look like, but like with our system here with FGF21 after stimulation, this is again a video. I just pulled a single frame out, but what you don't see in this system is that these green spots, those are lipid droplets. And what happens is over time, they just break away and disappear. So you see the fully differentiated one has lots of lipid droplets. You stimulate with FGF21, which is supposed to increase the release, increase glucose uptake, increase ATP activity. And I just in real time, that one at the top that looks like it's breaking in two was one while I was taking it. And then at the end, it broke apart into two and then basically disappeared. So yeah, you can collect full video as well on this.
All right, any other questions for Nick today? This is really cool seminar, Nick. Thank you for this.
Thank you. Like I said, the software, the SimFCS, there is a free legacy one available.
Mm-hmm.
And it's again, my mentor Rico, he coded it and made it back in 80s, maybe 90s, and he still codes. I don't know which program he used. It is definitely not Python or any of the newer ones. I've seen it when he opened it. It's pretty rough, and he's always willing to make changes to it too when people need it. So he'll do it right on the fly. So, but again, I don't even try to, because it's like over 150, 200,000 lines of code, and there's a lot of because he doesn't check up on it, and he makes multiple versions of it. And so there's a lot of different versions that I have where you click something and it just completely crashes the system. I'm like, what did you do? It just went somewhere else. And I have to go and tell him like, hey, this is just crashing. He's like, oh, you don't need this one. You need this other one version here. Like, okay, great.
Well, that's what we hope our students are learning, is about version control and a bunch of other things in GitHub.
It's super, super important to keep it up to date and stuff like that. And again, another example with Enrico is like he's so busy sometimes he doesn't get back. He's like, I emailed him, it's like, hey, I need help with adding this specific parameter to it. And he's like, okay, I'll get to you 6 months later. I'm like, he didn't do it. And then I checked online and he wrote a tutorial on how to code it in. I was like, oh, it's like, why didn't you tell me that? I was waiting for you to get back to me. But yeah. Keep them up to date, keep track of them, throw away the old ones. They don't crash a computer.
All right, well, any other questions for Nick today before we move into our workshop?
I got a quick question. Am I allowed to stay online for the workshop?
Absolutely.
Okay, great. My students
very much so. Yeah, please do.
Okay.
Okay, well, with that, let's give Nick a virtual clap, or turn on your microphones and actually clap out loud. And okay, Nick, you can now turn into a participant like magic.
Thank you.
And we will move on to our next event. Thank you so much. It was really cool.